Malhotra from Oregon Health and Science University. He will be presenting on molecular imaging in drug development with a focus on introduction to multimodality molecular imaging. This session will take us into the exciting world where chemistry meets imagination and innovation.
Good morning. All right, so far, so we talked about, experimental therapeutics, what all it takes to be part of experimental therapeutics. It's a multidisciplinary process, as you just saw. There are many, many disciplines involved. Now, when we do an assay of just petri dish. So basically, you take a drug, a small molecule, whatever you do, you're just studying biology. You have to know that it is correct or not. So previously, going back to, there were simple chemistry methods that came to understand the method that was in 18th and 19th century. Then came instrumentation method like chromatography, NMR, IR, all these to understand the molecules. NMR, I might have mentioned, NMR led to MRI, magnetic resonance imaging, जो hospitals में करते हैं, that came out of NMR. NMR came out first, and that technology then led to MRI. We use various techniques, we use various methods, and in order to make sure that we can follow what we are interested in. One of the techniques, for example, when the optics physics became more and more advanced, imaging, that when you light various rays of wavelength, when you light on something, it reflects depending on where the angle is, all of that you can measure and can get sense of the target. Imaging similarly is a modality which is very, very important now for the last few decades, and especially more and more advanced now, where imaging is used to be able to see कि क्या हो रहा है, whether it's inside your petri dish when you do, because for example, as I was telling you, I showed you on this side that you can make different cell lines. You can create cells, you can create vectors, because you have base pairs based on paper. For example, let's say if you have any cancer type that you want to understand and develop the new drug, new therapies for that. I'm focusing on therapy side because experimental therapeutic is more focused. The way we have designed this course is more focused on the therapeutic side. But you want to learn more about the biology. You want to understand what is going on inside whatever your system is, whether it's a cell, in vivo, or humans. Uske andar ho kya raha hai? So when you create vector, so I'm starting from a very simplified version. So if you have to create a cell line, you create cell line, and I was telling you what you do is at the end you can put a fluorescent, luminescent, some tag to that, and then that tag will light up, and based on the intensity of the tag you can see कि उसके ऊपर कितना effect है. You can actually quantify. आपने ड्रग लिया, ड्रग से ट्रीट किया, whether it's 15%, 18%, 80%, 90%, based on the intensity. कहाँ से पता चला कि 80% है, किसने कहा कि 18% है, right? There has to be a way to quantify. Okay, that was, and you can actually image. Now you probably are, I'm sure, familiar with or have seen, when you go to physician, MRI is a technique. When you go to the physician, or physician सारा लेते हैं, X-ray क्या है? It's image, right? जो image लेते हैं, that imaging using X-ray as technique that has come out, right? So imaging has become extremely, extremely important process in order to see what is actually going on. उसके अलावा तो indirect methods होती थी पहले. Imaging helps to know if there is a drug is affecting, for example, if there is a tumor type inside, be able to image to see what is actually where the tumor is, so surgeon can take out that tumor. The surgery which is done first. If you're using radiation, if you're doing laser, be able to see वो है कहाँ. But how do you see that? So through imaging. So imaging techniques, various imaging techniques have become extremely, extremely important these days, right? Same way, now this is, I'm talking about patient. Now trace back, same thing inside animal. If you're developing something, you're trying to understand, you need to know what is happening. So imaging, and there are various modalities जो हैं imaging के basis पे have come out. So I will just quickly give you an introduction. So I have highlighted these, and as I was telling you, I have taken notes in such a way so that once you review that, you will have more information. So it doesn't mean that your text has, or you have to, just go through text, but because I think the plan is that I will give all my slides to Dr. Gurjaspreet Singh, and then you will have access to that. So when you see a lot of text on my screen, personally, my presentation style is I don't like text. कहावत आपने सुनी ना, picture worth 1,000 words. Picture conveys the message more, but sometimes it becomes necessary. So you'll see on my slides, some of my slides, उसमें text होगा. जैसे कल रात को बैठ के भी मैंने उसके ऊपर yellow किया कि you don't read the whole thing, but read the yellow part. That's the important part. So anyway, why and how these modalities are helpful. As I was telling you yesterday, I showed one of the slide I had was where you see thousands, just say pick a number like 10,000 compounds go through, and maybe one will see the light of the commercial world. It's an extremely tedious, multidisciplinary, and extremely expensive process. Any and every technique that can help us to cut down the cost, the time, the resources, the frustration is a positive step in that direction, imaging being one of those. So it's a very time sensitive. So what new strategies are needed? Imaging basically helps in characterization of molecule level, protein level, about the cell lines, whether animal, you can image the animal. If you're doing something, image. So when you do, you actually see whether cross कर रहा है कि नहीं कर रहा. Yesterday, for example, now blood-brain barrier, how do you measure that? You can image whether it crossed or not. You can image to see whether the effect of the drug happened or not. उसकी जो दवा जो है, if it's tumor, you can see the tumor shrink हुआ. How to measure that? How to before or after treatment? So imaging is an extremely so these are in radiology departments. I'm sure PGI में भी है. All radiology departments. This is a routine process. सुबह से शाम तक वही करते हैं. Scans करते हैं, scan को पढ़ते हैं, scan को समझते हैं. Then they do the measurement and tell के patient को क्या है, कितना असर हुआ, नहीं हुआ, कहाँ है body में, जो भी या organ में जहाँ है. So it is not just predicting the clinical observations, but based of that you can use that information even to understand the mechanism, how it might be happening. Because if you assume that there is something, it is happening certain ways. Now there are many, and I'll come to what is happening in my institute, for example. So it has, as you can just imagine, पहले कुछ नहीं था. You do the analysis. Once it's done, you use indirect methods or invasive methods. These are non-invasive. You don't have to insert something inside the animal's body or human body. These are non-invasive. They don't invade inside. So, and you can just take a picture. So therefore, these are extremely powerful, extremely helpful, and extremely important. And that's the reason they are routinely used, because patient को कुछ भी inject नहीं करना पड़ता, कुछ नहीं पड़ता, खिलाना नहीं पड़ता, पिलाना नहीं पड़ता, but you can just simply take the image उसके साथ में. And in most non-NCDs, non-communicable diseases में बहुत application है, whether it's a cancer, cardiovascular, neuro, Alzheimer में. There are not many methods available, unfortunately. Neuroscience is a very fast developing field and unknown. So again, point being, if you want to pursue a career, be an expert, just pick, as a chemist, pick any one of these areas and just follow. And before you know, there's ocean and ocean of knowledge and ocean of unknown. So you can pick up an expert धीरे धीरे करके in one field, and before you know, you'll become an expert in that area. Just pick one area. Problem, I mean, there is so much unknown when it comes to the biological creation. So the I mean, many these phrases have been used in the past. You pick one protein, it will take you end of the life. एक प्रोटीन को समझने के लिए आप अपनी सारी ज़िंदगी लगा सकते हैं, फिर भी नहीं समझ पाएंगे, बट उसके अंदर आप मास्टर बन जाएंगे, उसके अंदर आप नाम कमा सकते हैं, क्योंकि आप सबसे पहले करने वाले होंगे जो उसको बताएंगे. So there is so much to be done. So imaging, for example, as a chemist, pick one, imaging agents बनते हैं, and I'll come to that briefly. So again, you know, it's an imaging thing which is used in the drug development from early assessment of target, protein को समझने के बारे में, drug development candidates जिस तरीके से बनाने हैं, and of course phase trials में, phase 1 to phase 3, again, depending on that, radiology can take the image, and I'm again using this for either neuron cancer or cardiovascular जो NCDs हैं. This is a routine thing, and as I said, it's an expensive process, but imaging, जिस मर्जी चीज़ की, any new technique, any new modality that can help in better understanding is a positive thing. So here is summary of various modalities which are used. Optical imaging, सिर्फ laser, सिर्फ light उस पर डाल के, green light, you get the sense. So there are, each technique, for example, has strength and also have limitations. Some, but strength can be used for whatever your goal is, whatever you're understanding in. Optical devices are very helpful in for example, high throughput scanning. So in my lab, we use very regularly. You take, when you do high throughput scanning, when you screen thousands and thousands of compound against a single protein to see कि कौन सा compound काम करता है, when you treat with that, you take the cell line, cancer cell lines, you can and put it in 96 well plate, 384 well plate, or 1536. एक ही plate के अंदर 1536 wells हैं. You can put your cells, you can put 1536 drugs at a time. So we, for example, if anybody has to do like 250,000, 2,50,000 के करीब अगर टेस्ट करने हैं, फटाफट प्लेट्स में ट्रीट करते हैं, then imaging, optical imaging can quickly help to see कि उसका असर कौन से वेल में, कौन से कंपाउंड ने कितना किया, and then from that large collection you can pick कि उसमें से कौन से hits हैं. So imaging is used there. Magnetic resonance, easy, advantage is clinical translation, but it's costly process. Ultrasound is used commonly, used again, clinical, it's a low cost, but it depends on the operator. There is a depth only up to which that UV radiation can go. अगर डीपर इनसाइड द बॉडी अगर करना है तो वो उसको पेनिट्रेट करके वहाँ जाएगा, नहीं तो यू कैन नॉट यूज़ दैट. यू कैन यूज़ इट फॉर द सरफेस के आसपास तक ही, राइट, बट सरफेस के आसपास तक है, डिपेंडिंग ऑन उसकी यूवी रेडिएशन कितने तक जा सकती है, उसको आपको रिफ्लेक्शंस तो देगा, बट दैन द एडवांटेजेस कि वहाँ तक जाता है, डिसएडवांटेजेस कि उससे डीपर नहीं जा सकता, तो you use as is needed. PET imaging, very commonly used, is costly, but it is one of the most commonly used imaging technique by radiologists. SPECT, another form of so PET is positron emission topography. If you see that, and then CT is again high spatial resolution, but it's not specific to target. It can give you full spatial resolution, but not specific to a target. So each modality has its own characteristic, which can be used, which can be employed for I'll just change the mode. Just give me a second. It is not in the presentation mode, and I'm trying to see how I'm trying to put in that presentation mode. No, it's not in the presentation mode. Huh? यहाँ पे view, under view, it should show here. Side master presentation view. Let's see. जो है when I do this it should
Here it should be the notes bar.
it should be the notes section. Yes, now it's better. Thank you. Okay, so okay, so this is again, you know, in as I was telling you, many ways by you where imaging we do this routinely. For example, all those who do a lot of in vivo work, we use this, for example, routinely. When a mouse studies are, for example, done, you inject whether it's a green GPF, green protein, or to tag as the optical imaging के part के लिए, and then you can from there also your distribution of your drug inside where it went. If it didn't went, you can simply take the images of that. Now, this is what I was telling you. You can do actually even mice high throughput. There are ways to do where you can actually just tell them. This is an example of a receptor. So if you want to identify a receptor or a protein, so receptor, let's say उसके right flank thigh में you inject the cell line, and you are interested to see in that tumor part what actually is happening. You can use imaging as a technique to learn more about various different, you know, ये जैसे receptor ligand interactions, whether it happened or it didn't happen. So this is tumor again. In the tumor size, to see what is the tumor size, you measure the tumor size. Now there are multiple advantages, as I mentioned, for surgeon to be able to see where to see the surgery, how much portion and what to do surgery. For a researcher to be able to see before and after what happened. If you're treating, is the tumor shrinking? What is the size of the tumor? What is the volume? You can measure you can measure all of that, and if it is shrinking or not, it tells you. So imaging can be used rather than having to just poke it out and take it out. Instead of having to euthanize the animal, you can actually, through imaging, keep on constantly giving your treatment and see how much and what the effect is happening. So again, imaging, again a powerful. Now clinically, as I was telling you, for clinically, there are many applications for identifying if there is a target or not, for distribution, if the drug is given or if something is happening, how it is distributed in various organs, whether it goes to the liver, intestine, stomach, heart, lung. You can take the imaging and you can just see whether the drug was distributed or not, instead of having to poke and take it out any pieces. Simply just the image, and you can follow that. You can put a tag and you can follow the tag and see by simple imaging it can help you. Pharmacodynamics, what happened, what drug does to the body. Remember, pharmacokinetics is what body does to the drug. Pharmacodynamic is what drug does to the body. Again, shrink hua, nahi hua, kitna hua. And again, patient selection, patient stratification. Even there, imaging is used to see whether the patient is the right patient or not for the treatment. Now there are, how do you do that? So basically for positron emission topography, there are, now how do you do that? As a chemist, think for a second. Aapka neutral compound hai, does not have anything, so how do you do that? What will give the signal? So there are radioisotopes. Oxygen hai, to instead of giving oxygen 16, you can give, as you see here, oxygen 15, carbon 11, your gallium 68, fluorine 18 is one of the most commonly used compound in the world is F18. So in our university we have our own cyclotron, so I oversee that center also, Radio for Pharmaceutical Research. So what we do there is we design tags, we design for many our researchers, whether they're in any field, cancer, neuroscientists or cardiovascular imaging, jo bhi jahan bhi chahiye, and even monkey colonies, they all come to our lab, they all come to our center, whatever they need. So if they design a ligand which they think is actually affecting, now what you need to do is you need to add one of these tag, fluorine, 18 हुआ, gallium, oxygen, carbon, you attach to their molecule of interest. They inject, they're simply through imaging, then they follow. Okay, treatment is happening or not. So this is a huge field now. It's a very big field. So again, it's purely chemistry. So in my radiopharmaceutical center, I have a radiosynthetic chemist, and it's a highly demanding, they are very much in demand because there are not that many. So if you want to become a radio chemist, demands are high. And I pay them 3 times more than I pay to my chemists, because they have to be licensed. They take their special license, but then they are highly in demand because nobody knows how to work in the radionuclide. These are radionuclide. But anyway, these are the applications. So you tag whatever the molecule of interest a scientist has for whatever reason, for understanding that, for the treatment part, for simply understanding the mechanism, understanding the biology. But so the scientist comes to us as radiochemists and says, this is the molecule I'm working on, this is the antibody I'm working on, this is the RNA I'm working on, or antisense I'm working on. It works in something. Now I'm interested to see how much it in fact has inside the animal. But I want to do very quantification. So our radiochemist will take that, whatever the molecule of interest, they do the whole synthesis process, but in somewhere in the synthesis process, they will insert one of these guys. That's one part to understand. How to select, which one to select, is dependent on also on the half-life of the radionuclide. So if the half-life is only oxygen 2.1 minute, just imagine आपने बनाया और जब तक hospital पहुंचोगे उसकी half-life तो खत्म हो गई, right? So what you do is you make a large quantity of that. So by the time it gets to the, अगर आपने बनाया है 10 मिलीग्राम, हर 2.1 के अंदर वो 5 हो जाएगा, 2.5 हो जाएगा, 1.25 हो जाएगा, वो होता जाएगा. So you make a large quantity, and if that's the only thing which is visible. And therefore, for example, in our many places, it's like I said, there are many these radio pharmacies, as they are called, radio pharmacies, people. And then the carbon 11, 20 minutes, 20.4 is the half-life, right? Gallium 68, so that gives you still. So हमारा जो monkey colony है, it is about 25 minutes around away from our radiopharmaceutical center. So you make enough of that. We have a courier service ready. That's all they do. The courier service का full-time job है. When it's needed, they simply जाता है और एकदम lead उसके अंदर, because you cannot take the radionuclide outside. They have to be protected to the body. Put it inside, उसके बड़े jar होते हैं, lead-based jar. Put it in the jar. Courier rushes to the labs which are in the monkey colony. They do that, but we make enough of that. They can do the treatment. 68 minutes उसका half-life period है. And same way, fluorine. This is why this is, as I mentioned, FDG जो है is the common. If you Google, if you are saying FDG, FAD, this is the most commonly used. क्यों? क्योंकि उसकी half-life लंबी है. आपने बना के दूर तक पहुंचा सकते हैं. And literally, many times, depending on, so for example, we are south of Seattle and north of San Francisco के बीच में हैं. So depending on, so British Columbia, state of Washington, Oregon, California, and on the east side, then you have Idaho, Montana, दूर hospitals, and they want to do the treat. There are helicopter services. If that is what is needed, helicopter बनता है, फटाफट जाता है, helicopter will fly it there, take it to them, wherever the treatment has to happen. But all things, that's the service part. But to design, these are taken into account. Okay, it has enough of half-life to get to. So for patients, for example, they are, so we are, so before, I'm at Oregon Health and Science University now, but just before that I was at Stanford. And even though I do some work there, but these big universities, they have their own cyclotrons. Otherwise, most university they have to just get from जैसे आप vendors outreach वगैरह से लेते हो मंगवाना पड़ता है उनसे. So we have that advantage, that gives the leg up in the sense that our investigators can think of doing research in this area a lot. But again, these are things. So you can put any of these, and there are more. There is a whole list. I'm just showing you four, just to give an idea okay, how and what factors are logistically, practically important. उसकी half life क्या है देने के लिए. Same way there is nitrogen. Cardiovascular studies के लिए nitrogen बहुत is commonly used. Cardiovascular imaging के लिए. So our center, for example, is, if this is the center, associated building, attached building next to is the cardiovascular, and maybe जहाँ से enter करते हैं, not too far from here, maybe 100 yard, no, not 100 yard is too big, 50 yard is our hospital, 4-tier hospital. So idea is location is such so that you quickly it goes to for patient के लिए जाना, जिन्होंने patient treatment के लिए, physicians they need, doctors they need for the patient. So that goes there. Now again for that, there are regulatory environment कि कितना clean होना चाहिए. आप हैं, when we go inside the radio pharmacy, आप ऐसे नहीं यहाँ चले गए. You have to be like spaceship. इतना tight नहीं होता, but you have to be fully, because and you have to be absolutely हाथ साफ किए हो, अपने gloves पहन के you go there, because highly sensitive part, right? But this is how the field has changed tremendously. Now, if you guys are interested to get into therapy, radiotherapy is presumably going to be a new frontier in therapeutic drug development. And if you want to build your career, if you are interested in, think of radiopharmaceuticals. There is a lot of investment happening. You don't need to worry about that. There is billions and billions of dollars are being invested क्योंकि उस field को आगे लेके जाने के लिए. The idea being कि जो chemotherapy है उसके side effect हैं. Other modes of therapies, there are side effects. What if you can come up with a radiopharmaceutical? Radiotherapeutics, for example. So radiology भी करता है, and also it's a therapeutics. It can have a paradigm shift. But that's the field is evolving. So if you are at an early stage, think of doing your PhD in one of those areas. Those who are have done PhD in chemistry, think of doing your postdoc in those areas. If you show interest in, anyway, I think that certainly came to my mind. If you are really interested in doing that, apply to radiopharmaceutical labs. As a chemist, you apply that you are interested in that, because not many people will go into that route because everybody wants to go to pharma industry. But radiopharmaceutical के अंदर आप करते हैं, it's a very niche area, it's a very limited area, but the market is booming. And like I said, I paid 4 times more than I pay to my chemist. So scientifically, it's an extremely important area and rapidly growing area field of research. So I mentioned about MRI. So basically, weak magnetic dipole, you know how the NMR works. Atoms, electrons, how they are aligned depending on what their polarity is. It splits. You look at that, 3-dimensional, look at the J constant, look at the splitting, and you can identify. So similar principles are applied to imaging and for body imaging. And now, advantages. The body has aquatic water environment, so hydrogen oxygen bonds easier, not too difficult, and they are not interfering into the weak low energy. So you can easily determine the concentration of hydrogen atom, how much water or a fat, and their local environment.